Okay, hi everyone, and welcome to today's webinar. My name is Casey. I am the Community Engagement Manager for Mission MSA. Before I hand things off, I'd like to know that the information presented here is for educational purposes only and is not intended for providing medical advice. Additionally, we will be taking Q&A at the end of the presentation. And you can use the Q&A function in the bottom of your screen to submit those questions throughout the presentation. And yeah, without further ado, I would like to pass it off to Dr. Sonia Scholes, who is a physician scientist who specializes in neurodegeneration. Dr. Scholes?
Well, thank you so much for having me. It's a real pleasure to talk about a topic we think quite a lot, and that is how can we leverage modern genomic tools to understand very complicated neurological diseases, and multiple system atrophy really exemplifies that. Okay, I hope you can see my slides, but please complain if you don't. So before I get started, I am a employee of the National Institutes of Health. And so the presentation here is really designed to give you a little bit of an insight as to how we think as researchers we can use modern genomic tools and how that could help us advance both understanding of neurological diseases, but also treatment approaches. And so none of the things I say are official, government guidelines. So these are, the things that I have put together, and they're not yet ready for clinical applications, but they give you at least some insights as to how we think about this disease. And so a couple of topics that I would like to cover in this webinar is, first, I want to just talk a little bit about what actually is multiple system atrophy. I know we have different viewers on the line, some may be more knowledgeable than others. And then I really want to talk about, how do we actually think about genetic factors that may play a role in multiple system atrophy? Why do we even think there are genetic factors involved in a apparently sporadic disease? And also really want to highlight, how we can leverage modern genomic advances, technological advances in a more translational context. How can we gain more insights into the disease? But also we really want to obviously advance therapy developments to make this disease easier treatable and perhaps one day curable. And so let me just, start out with giving a little bit of background information. So multiple system atrophy is a neurological disease. It is sporadic in the community, which means there's no apparent family history. It's usually, affects individuals in their mid-50s, but there is some spread from case to case. And the clinical symptoms are quite varied from person to person. There's a lot of problems with motor control. So there can be, symptoms that look a little bit like Parkinson's disease, where you have rigidity, slowness of movement, maybe tremor, balance issues, or troubles with motor coordination. So the first one is called Parkinsonism. The second one are the cerebellar features, which are more a motor coordination problem. And then there are other symptoms that relate to the autonomic nervous system, which is the nervous system that controls our internal organs, so such as the intestines, the bladder, temperature control, heart rate. That's the autonomic nervous system. And there's a lot of variability to which extent different parts of the nervous system are involved, and that explains why not every MSA patient looks the same compared to another patient. And there's a lot of different signs and symptoms that can be quite confusing in early stages to piece together that they're all actually related to one bigger disease process. So it's a really challenging disease. We distinguish two major subtypes based on what is the predominant kind of clinical feature. So patients with more Parkinson-like symptoms are the MSAP patient subtype, which we see more in European ancestry individuals. Whereas the patients who have more motor coordination issues are patients labeled as MSAC for cerebellar variant. And we see that more often in individuals of Asian ancestry. But the disease is one that, for unclear reason, comes up with no apparent cause. It usually affects people in their 50s, so it's an adult onset disease. And despite our best efforts, it is progressive. Most people are wheelchair bound after about 5 years, so it really is an aggressive disease. It affects both men and women roughly at the same rate. And at the present time, we do not understand what's actually causing it. There's more and more thoughts about risk factors that are playing a role, and we'll specifically zoom in on the genetics. But because of it, because we don't fully understand what's going on, we actually only have symptomatic treatments available. These treatments do make a big difference, but none of that are actually stopping the underlying disease process or slowing it down. And so it's a huge unmet need trying to tackle this disease by understanding it better and really trying to stop the disease from its progression. And so the current standard of care is actually quite complicated because there is no symptom that is specific to all patients, and there's no problem that's really specific to everybody. There is no single kind of therapy that fits all, and it becomes quite challenging trying to piece together the right kind of drugs that could be tried, but also non-pharmacological therapies. There's also from healthcare challenges. Not everybody has access to institutions that offer all of the services. But increasingly, we all realize that in order to manage this disease effectively, even the standard of care needs to be multidisciplinary by design. I really want to also highlight that there are these MSA centers of excellence that really provide the standard of care, but also are a big important hub for recruitment of patients into clinical trials. And Mission MSA actually has a really nice kind of resource on their homepage where they describe these MSA centers of excellence where people can access the care. But what is actually happening in the brain of patients with MSA? So we have learned that for unclear reasons, there is deposition of a protein that's called alpha-synuclein. This protein is usually, we all have it. It's a usually unfolded protein. It's important in neuronal plasticity, so it's important to help with signaling between nerve cells. And for some reason, it becomes misfolded and starts to build up in the nerve cells. It's also interesting that it's not the only disease where we see alpha-synuclein deposits building up. There are other diseases such as Parkinson's disease and Lewy body dementia where synuclein is building up. And we summarize these diseases under the umbrella term of synucleinopathies. The difference is that if we look under a microscope at the brain tissue, Parkinson's disease and Lewy body dementia have buildup in neurons and then actual nerve cells that we call the Lewy bodies. Whereas in multiple system atrophy, we see the main pathology happening in a type of cell called the oligodendroglial cells, which support the function of neurons and also build kind of an insulation around the neurons. And in those individuals, we do see these so-called glial cytoplasmic inclusions, a bit of a mouthful, that are made up predominantly of alpha-synuclein protein. And recent studies have shown that although they are made up of the same kind of synuclein protein, the misfolding is different in multiple system atrophy compared to the other synucleinopathies. So there are some molecular differences. And so while we can distinguish the disease quite easily on pathological grounds, and when we look at brain tissue, it can be quite challenging to tell the diseases apart on clinical grounds, particularly in the very early disease stages. And so there have been, major advances already now due to, modern technologies, genomic sequences, data sciences, to gain insights into Parkinson's disease and Lewy body dementia. But MSA research has really been quite challenging, and it's not for lack of interest. It's because it's a very challenging disease to study. So first and foremost, it is a sporadic disease, so it doesn't typically run in families with a few extremely rare exceptions, which I'm going to talk about. It is also quite rare. So on average, we estimate that there are about 10 to 15,000 cases in the United States. So it's still, hard to get large cohorts of patients together to study the disease. And usually for many of, especially genetic research studies, we need hundreds, ideally thousands of patients to really, tell the noise apart from what is a real disease-associated genetic change. And then the diagnosis is still sometimes challenging. There are a number of neurological diseases that we have learned about over the last few decades that can sometimes really look like multiple system atrophy and can actually mimic it. And so you can imagine if you had to do a research study or a clinical trial, for example, and a substantial proportion of those patients may not actually have the disease, and that could potentially lead to false signals or to false negative results. And so we have to do better in trying to diagnose patients very early on and tell them apart. And this is in part where we use modern imaging technologies, but also genetics can really help us to stratify those patients. And then the other challenge is that the symptoms can be quite variable from person to person. Sometimes it's more obvious what we're dealing with very early on, but in other cases, the diagnosis is often very delayed, where patients are already so impaired and so much struggling with the disease that research participation is just too much, and it's perfectly understandable. So we need to do a better job in really recognizing the patients and working with them early on. And so I'm a geneticist who is studying these neurodegenerative diseases, and I'm gonna just highlight how we actually approach these conditions. So then the very first question that I also get from a lot of patients in the clinic is, is genetics actually playing a role? And the answer is, it's certainly there is some evidence that there are mild genetic factors in the background. It is extremely rare that MSA is running in families. So genetics is probably not the cause of it, but genetics are still influencing it. And even if we are studying these milder genetic factors that may explain, why somebody may have an earlier onset, why somebody may have a more aggressive disease course versus another, it gives us at least a molecular anchor where we can look in the cell, we can leverage that for modern disease modeling, etc. That doesn't mean that genetics is the answer to multiple system atrophy. There's good evidence that there are other factors that clearly play a role in the underlying disease pathogenesis. There may be lifestyle factors such as, diet, physical activity, exposure to, certain pesticides, etc., that can contribute. But there's also evidence that there are so-called epigenetic changes that are happening, which is not just looking at the genetic code, but what's on top of the genetic code that is influenced by the environment. And it's really the combination of all of those risk factors that are probably giving rise to the disease. But for now, I'm just going to zoom in on the genetics and just to see how we can actually pinpoint certain kind of sensitive areas in the cell. And so to give you just a quick primer on genetics, so when we talk about the genetic blueprint, what we're really talking about is DNA. DNA is the molecule that essentially encodes our cellular machineries, the type of proteins that we produce and how they function. It's a pretty elaborate kind of code. It contains about 3.2 billion letters for a human genome. And the letters are actually, there's 4 different type of letters, so-called nucleotides, that are arranged in various combinations. And it's quite amazing. There's a lot of similarity between humans, but there are also variants, subtle differences that explain, different traits, why I'm tall, what, my eye color is, etc. But it also has, it explains why some people look different, why some people have different responses to treatments, and it's important to try and tease apart how genetic variation is actually playing a role in human health and disease. And so we are in a different era nowadays. We are really in the genomics era. The first human genome, which was published in 2001, took about over a decade of work, hundreds or actually thousands of people working together, $3 billion of investment to just put one single genome together. And now, because the technologies have advanced so much and the computational capabilities have advanced so much, we can actually do the same thing overnight in my lab. So it's the growth of knowledge on the genomic side is not just a linear one. It's really been an exponential growth. And so the challenge really for us as researchers is try to really get through the genetic information, tease it apart, and how it actually plays a role in various neurological diseases and other diseases. And so there's untapped opportunities there to learn something about very challenging diseases that we haven't been able to tackle beforehand. And so I would split kind of applications for genetics into two different sections. So the first one is we can actually use genetics in the right kind of context to help us with a differential diagnosis. So as I mentioned early on, some people can have a neurological disease that can really look like multiple system atrophy. But if there is a family history of other members with neurological diseases, that could be a clue that perhaps we're dealing with a so-called mimic syndrome or a lookalike syndrome. And increasingly, because we have modern genomic technologies at our disposal, we can actually already test for them. So those are diseases that are falling under the umbrella term of spinocerebellar ataxias, familial Parkinson diseases. And it's still, we learn more, every year we identify novel disease causes, but the more common ones we actually can screen for. And so with the availability of genetic counseling and genetic testing, sometimes we can actually establish a molecular diagnosis and can say, yeah, this is actually not MSA, this is a mimic syndrome due to a mutation in this and this gene. And that may be useful in the clinical context because it allows us to refine our counseling. Not every disease has the same kind of disease course as multiple system atrophy. There may be other complications. There may be other treatment trials that might be relevant for these so-called mimic syndromes. And so it's important to recognize them. And we can use genetics for that purpose. But then the other application is that we want to use genetics to understand what is actually going on with multiple system atrophy. Ideally, we want to learn what is causing it. It is unlikely that the genetics is the cause of it, just because we don't really see it a lot in running in the families. But we think that there's good evidence that there are actually risk genes that are playing a role, that there are modifiers that are playing a role that may explain some of the variability that is really going on. And by pulling together a lot of information from large cohorts that have been ideally collected over different countries and with very high standard of certainty, by using a lot of additive diagnostic tests such as imaging and biomarkers, we can actually gain a lot of insights into this disease. And so the ideal scenario is that using a genetic tool, we pinpoint a genetic variant. Once you know where to look, you can already start to make some hypotheses. You may know which protein this particular gene that carries a variant is coding for. You may already know what pathway it is involved in, and then you can test that. You can take it into a model system. For example, you can generate stem cells just out of a blood sample these days. We can generate nerve cells out of a blood sample, which is quite exciting. We can generate mini organs out of stem cells, or we can treat animals and examine them by really looking at these particular genes. And the hope is that by really knowing where to look and manipulating those genes and pathways, we may be able to come up with therapeutic strategies to really correct that particular genetic defect. And then ultimately, we want to bring it back to the patients and stop the disease, or at least slow it down. So that's the ultimate goal that we have with our genetic research efforts. And so we have several different tools in our toolkit. I just want to highlight a few. So the traditional approach in genetics has been, to look at familial cases, which is, familial diseases such as sickle cell, anemia, Huntington's disease, where it's very clear family history, every generation may be affected, etc. Now in MSA, that's been very challenging because it's extremely rare that we see familial multiple system atrophy. And as a consequence, for the most part, what we are using as a tool to identify genetic variants is so-called association tests. And the basic concept is that you need to, recruit a large cohorts of patients who are affected and a large cohorts of control who are healthy from the same population, and then you compare them, cases versus controls. You can do that on a gene level, but you can also nowadays doing genome-wide. So you can look at all 20,000 genes and all non-coding regions in one go. You have to do some statistical corrections. And then what you're looking for are significant differences, either variants that are more common or less common in the patients. And that at least clues you in as to where you need to look in the genome, where is something that's different, and what are the genes in those regions, and then you really try to work out step by step what it actually does. How does it affect the pathway? How does it affect the transcripts that are coming off and the proteins? And ultimately, how does it play a role in the disease process itself? And so we can map this out on this so-called Manolio plot, where we look at the genetic architecture of a population with a given disease. So on one hand, we look for, in the familial cases, for mutations that really are of a major impact. If you have that, you get the disease. So in MSA, that is extremely limited. There's a little bit of work done in that space, but because it's really mainly sporadic, we are really in this space here where we look for associations, and those association tests really give us an insight into risk genes. So not the causative genes, but risk genes. So those are genes that are not enough to cause the disease, but they're playing a role in a statistically significant manner. And while it's a bit frustrating when we talk about risk genes, it's not something that we would ever put into a medical report, etc. But knowing these risk genes can really inform research and really can tell us where we actually have to look in order to develop the new treatments that target the underlying disease processes. And so I'm going to just show you some of the major studies that have been done in multiple system atrophy. As I mentioned, MSA is extremely, extremely rarely found in families. It's so rare that if there is a familial occurrence, it's worth writing it up as a publication. And so there are a couple of families that have been described in Japan. It's actually the most families that actually have been reported actually came from Japan, where you see patients where there's one generation that appears to be affected, whereas the parental generation is relatively spared. And when we see that pattern, that's often a fingerprint for what we call a recessive inheritance, where you need more than one spelling mistake or mutation. You need two, both from the maternal side and the paternal side, coming together. Whereas another family that's been described in Germany has seen, two subsequent generations, both affected with multiple system atrophy and also confirmed by autopsy. So sometimes, the question is it just bad luck? It can happen. Or is there actually a reason why these individuals really have disease? And at least in the Japanese population, we have at least an answer for it, that in very, very rare cases, there are spelling mistakes or mutations in a gene called CoQ2 that are playing a role in multiple system atrophy. So what you see is not just one mutation, you actually see two because it's recessively inherited. And we actually don't see these type of mutations in any of the European ancestry cohorts. We've really been looking very hard. So it seems to be very much, focused in on the Japanese population where this has been described. But even if you don't have two mutations, if you just have one of them, there is a common spelling mistake that changes one letter of a protein that seems to affect the risk for multiple system atrophy in individuals of Japanese and Southeast Asian ancestry. And again, there are real population differences. We actually don't see it in the European ancestry population. And so it's important that we, as we study patients with these diseases, that we have to think about it, how does it affect different populations? But the good news is there is actually a real glimmer of hope. There's been already a clinical trial that has leveraged this type of knowledge and basically has been given a prodrug of CoQ2 as a supplement. And it was, well tolerated, it was safe. There's even some evidence that it may have been beneficial. It's been shown in individuals of East Asian ancestry. We don't know if that may also hold true for individuals who are of European ancestry, but at least you already can, appreciate that there is a lot of effort to try to really focus in on some of the molecular defects that are going on in MSA. And then there's other insights that have come out of related fields. The genetics of Parkinson's disease is actually much further along. It's a much more common disease. And we've learned over the last 2 decades that sometimes, there are, known familial forms of Parkinson's disease. There's one familial form where you have spelling mistakes or mutations in a gene called SNCA. This particular gene is, curiously, it encodes for the alpha-synuclein protein that I've shown you actually is building up in multiple system atrophy. And although when we screen for these exact mutations in our multiple system atrophy patients, we don't actually see them. But curiously enough, when you look at the brains of these patients, in addition to the Lewy bodies that we would expect in Parkinson's disease, we also see these oligodendroglial inclusions that really look like the glial cytoplasmic inclusions that we expect in multiple system atrophy. And so it really suggests that there is a molecular link at this particular synuclein gene. And it's been really leveraged to do more research. And perhaps there's also an interesting insight. Perhaps the reason why we don't actually see mutations in the blood when we screen patients is because, not all of the cells in the body carry this mutation. There's, a small study from the United Kingdom where they have looked at brain tissues and they found that MSA patients often carry an extra copy of this SNCA gene, but only in some of the brain cells. So you could imagine I can screen your blood as much as I want, but the problem is actually only the mutation is only present in brain cells. Now, we don't know how frequent that is across the MSA patient population, but again, it really highlights that we really need to focus very closely on the SNCA gene. The SNCA gene is also at the core of MSA models that have been developed. So for example, there are mouse models where we can overexpress this particular gene. We can coax the oligodendroglia to express more of the alpha-synuclein protein. And when you do that artificially, what you get are inclusions that look really like the glial cytoplasmic inclusions that we see in patients. And also these animals have a lot of the problems that we do see in patients, such as having problems with their motor control. And so a lot of the studies that have been going on, are still going on, are based on using these particular transgenic animal models. So the genetics has actually been quite helpful in that regard, as a way to study what's actually happening. And then, but these are really the familiar ones. These are exceptionally rare. But how about the actual risk genes? How do we go about it? And there is evidence that risk genes are playing a role. So we can do so-called heritability studies, which is where we estimate on average, is there a difference in the genetic makeup in MSA patients versus controls? And there is, evidence that there's at least a mild genetic contribution. We also see that there's a lot of variability from patient to patient in terms of age at onset, treatment response, progression of the disease. For example, we see more MSAP cases in European ancestry cases, and more cerebellar subtype cases in Asians, which may suggest that there are differences in the genetic background that could explain that. And we have definitely seen, a lot of interesting insights into risk genes by studying, large cohorts using genome-wide association studies. And so I'm just going to talk about a study that we recently led here at the NIH, which was a major initiative where we wanted to do whole genome sequencing of large cohorts of cases versus controls, and large by MSA standards is still modest, but the more the better. We were fortunate that we had over 800 cases and maybe 7,000 controls. We built a large collaborative effort of over 20 sites across North America and Europe to really bring these cases together and do a genome sequencing study. And we're particularly fortunate that a lot of the brain banks collaborated with us, where we definitely know that this is what the disease is, that we don't have a lot of misdiagnosed cases, but we also included patients where we have pretty good, at least clinical evidence. Those are the probable cases that we are really studying MSA, that the risk for including mimic syndrome is very minimal. And so all of these individuals have undergone whole genome sequencing using a standardized pipeline, sequencing using the latest technologies, and then putting these data together on a cloud environment, and then analyzing them to gain some insights. Now, the objectives of the study were that we apply modern gene discovery tools, and we've already done some of the analysis, but these data are still being used because genomes, I said, are incredibly rich data sets, and there's many more analysis that can be done. But the other important goal was that we generate a genomic resource that can be shared with credible researchers. So when I say sharing, is we upload de-identified genome data into so-called knowledge portals. So where somebody from any part of the world can apply and then work with these data, combine the data that we have with their data, and then gain new insights and really drive up our likelihood to discover new things that are important in this disease. And so it's just as a major highlight, what we actually found was we did a so-called genome-wide association study where we screened over 9 million variants across the entire genome, and we looked for significant differences. And we indeed identified 4 different regions in the genome that are different in MSA patients compared to controls. And so I'm just highlighting this, what we call the so-called Manhattan plot, where each dot represents one of those variants, and we see all of the variants showing up, and what you look for are these so-called skyscrapers. These are the significant signals, and we can see 4 different signals. Now, these signals just tell us something in this area of the genome is different. It doesn't entirely explain what exactly it is. There's often multiple genes in this region, but sometimes using other statistical tools and other types of data, we can really pinpoint what's the most likely gene that is driving risk. And so we were able to actually prioritize 3 of those genes They have these acronyms, these names, but they tell us something about the underlying disease process. And so one is important, especially for protein handling. And then this KCTD7 gene is also an interesting gene. It's a gene that actually has been implicated in neurodegeneration previously. There's a rare neurological disease where you have complete loss of that gene, where children, very young children, have this really devastating neurodegenerative disease. In multiple system atrophy, we see that there are decreased levels of the transcript that are coming off that gene in the oligodendroglia, which is exactly where the pathology is happening in multiple system atrophy. We know that this gene has another important function. It regulates a group of proteins. We call them calpains. Don't want to make it too complicated, but it's basically important for determining which proteins need to be discarded, and when a cell needs to go into cell death mode. And it's an interesting target for therapy development. So there's even been interesting mouse models that have targeted and have corrected that and may be protective. So there's a lot of hope by just knowing where the problem is, we can start to really come up with new therapies that are really targeting an underlying risk gene. Okay, so when we look at the genetic architecture of multiple system atrophy, it's still very limited what we know, but there's hope. And we've seen a lot of acceleration now because we have more of the tools available, we have more of the technologies available, but we cannot do it without our patients. And we need to drive up the numbers in order to identify a lot more that has not yet been described, or that we haven't yet really understood. And so that really brings me to the last kind of section of my talk, which is, how can we go from, pinpointing these kind of interesting regions in the genome to how can we go from that to actually new drugs? And so one thing that we immediately can learn is, when you know which genes are involved, you can actually study them. You can pinpoint what the underlying pathways are that are involved, and what are some of the molecular abnormalities in the cell. And so some things that we have learned are, for example, there's some problems with responding to stress, oxidative stress, for example, which is, our cells are very active and alive, they need energy, and if there is a discrepancy between the demand and the need, that can cause cell death. But there's also problems with protein building up. So synuclein protein is misfolded, and for some reason the cells no longer are able to actually break it down and get rid of it. The cells are very complicated machineries. There's a very fine balance between the proteins we produce and the proteins that we discard. And if as we age, that balance is anyway a little bit off. But if you have certain genetic risk factors, that can really be a reason why you may be more susceptible toward developing disease. And then there's important metabolic factors that could play a role in how the cells are responding and how they're able to manage certain stressors, a disease process, and perhaps other reactive factors. There's also neuroinflammation, which is certainly seeming it also seems to kind of exacerbate some of the symptoms. And so each of those insights and those pathways give us ideas to how we could potentially tackle the disease by targeting these pathways, either in isolation or together, and really try to calm down the disease process and delay the progression of the disease. And so there's really a lot of hope that the genomics can inform drug development, and there's good reason for that. So other fields are a little bit further along in terms of the genomic characterization. Those are fields where we have a lot of patients. So patients, for example, diabetes research, there's millions of people, and many of the genetic insights have already resulted in new drugs, just because we know where the gene is, we know the pathway, and you can bring it into cell models, animal models, And many of those drugs are really now very successful and are already changing survival for many patients. And we hope that we can do the same thing for a lot of these neurodegenerative diseases, including multiple system atrophy. And the other thing that I would like to mention is that, many of the drugs that really have genetic support are also the ones that are really doing well in the clinical trials pipeline and ultimately in the drug approval process. Because if you're really targeting the underlying molecular deficit, it's much more likely that you're going to be successful. And so it's worthwhile doubling down on our genomic efforts to really pinpoint the underlying molecular problems. And so the path to getting toward precision neurology and precision treatments is really by starting to tease apart the molecular knowledge. We've made some real progress. There's loads more work to be done. And while the genetics is important, it's only one part of the puzzle. But we can really already leverage the genetics to improve our diagnosis, sometimes by just ruling out other mimic syndromes. But we can also highlight certain biomarkers that may play a role and really become smarter in how we do clinical trials, but also which drugs that we should use. There is increasing, knowledge about so-called drug repurposing. As we have seen from various genetic research efforts, many of the pathways that are showing up are actually not unique to one disease. So, for example, I mentioned, protein handling pathways are often impaired in many other neurodegenerative diseases, such as Parkinson's disease. And you could imagine that one drug that works, let's say, for Parkinson's disease could immediately also be applicable to multiple system atrophy, because we know there is a problem. And so we can really accelerate the drug development pipeline, be smarter about how we treat this disease. And so there's some challenges that we'll have to tackle as a research community and as a healthcare community. We need more patients to participate in various research studies. So we need larger cohorts, ideally thousands of patients, to really drive up the numbers. And that's where the genomic tools are really powerful. We can also, use genetic data to correlate them with certain clinical features. But in order to do that, we need very deeply characterized cohorts. We need ideally patients who are followed longitudinally over a longer period of time, so that we can see which genes are affecting the disease progression, the treatment response, etc. And many of these data are still relatively scarce and still need to, we need to build them up very rapidly as a research community. And this is not research that one person can do or one center. This is really researchers from all around the world coming together and developing these data sets. We also, we have learned that ancestry makes a difference. Not every patient has the same risk profile in every population. And so there are differences in the clinical presentation, for example, in Asians versus European ancestry individuals. And we know very little about, for example, African Americans and Africans. And so there's much more work that we need. And so we need to really engage with the research community. And ideally, we want to create foundational data resources that can be shared, that people can access. And so this is where we are very passionate. So my lab has really been at the forefront to share the genomic data that we generate. So all of the genomes that we have sequenced, we can, you can access them as a researcher, and they are on various data portals, and others will come, and we hope that others will continue to add their data sets so that we can build up our knowledge as we go along, and hopefully as a research community really tackle this disease and figure it out. And so, I encourage patients, if they're interested in helping to participate in clinical trials, there's a great database called the clinicaltrials.gov portal, where clinical trials are, advertised, what they're doing, what they're looking for. Some of them test new drugs, some of them are more for studying the natural history and the biological changes that are occurring. And each one is important. we also need to really speak up and advocate for the MSA community, because advocacy really makes a difference. It has really been a major driver for many other diseases. So for example, Lou Gehrig's disease has had, a lot of success with getting extra research support. There's a actual research program that was recently released that gives extra funding for the ALS community. The same has been done for Parkinson's disease, and we really need to speak up and request those resources for our patients. And then we need to raise awareness and educate the next generations of physicians, scientists, healthcare providers, patients, so that we really recognize the disease early on and get patients into the right kind of treatment centers and clinical trials. And so some key messages that I was hoping to convey in this webinar today is that, yes, MSA is a sporadic disease. And, in those sporadic cases, risk factors are mild, but they are playing a role. We do see rare, extremely rare familial cases, and we haven't explained all of them. So there are potentially more genes that could be found. There's a couple of the genes that have already been implicated, and each of those give us some clues as to where the problem may lie. And we're already studying them either in clinical trials or still in model systems, and hope that it will give rise to additional clinical trials that really target the underlying problem. And so genetics is really a starting point for understanding a disease. It's not the answer to all of the questions that people may have, but it's a very important anchor as to where to even look. And I hope that ultimately these molecular insights will facilitate therapy developments that are not just treating the symptoms, but are actually delivering the cures that we all are looking for. And so with that, I'm gonna stop here, and then I'm happy to take a question or two that may have come up. All right, I see a few questions in the chat, so I'm gonna just review that. So there have been a number of risk genes already studied in MSA, and the results for many of those risk genes are variable. So there's one question specifically about NPC gene, which is a gene that causes, when mutated, a different neurodegenerative disease. But it's important that we don't see it just once, it's important that we replicate it. And so it is an interesting observation, but I think the question is still out there whether this is a real MSA gene or not. NPH, this is another gene. This is another question here. NPH, which stands for normal pressure hydrocephalus, has been seen as mimicking disease in families. So this is, NPH is where essentially there is buildup of fluid in the brain, and sometimes that can cause a lot of symptoms such as slowness of movement, but also bladder symptoms, cognitive issues. It's usually something that we can pick up on imaging, and so we can distinguish that from multiple system atrophy. And so, those are important decisions in the clinic. So when we first see a patient to consider these differential diagnoses. So it's not just genetic differential diagnosis, but it's also other diseases such as NPH that need to be addressed, but they're different from it. It's a good question. And I see another question here, which basically asks which medical institute has leading research on MSA, any support network for MSA patients, what other resources are available for MSA patients. And so that's a great question. So there are multiple centers that really have been focusing in on multiple system atrophy. There's not just one, there's a whole network of centers of excellence. And so I really would like to encourage you to check out the Mission MSA homepage that lists those centers of excellence across the country. But also, there are centers of excellence in Europe, so it's not just one center. The support network is often as part of this of this particular Center of Excellence network, but also, please leverage online resources such as the Mission MSA portal. Cure PSP also has a lot of important online resources and online support groups that you should look into to really, learn more about the resources that are available in clinical trials. Okay. Let me see, there's a couple of questions that are coming in. I'm just trying to see which we should tackle. Some of them are pretty much the same. And so one question that I do get, and it's a fair point, is what are the environmental factors? And so, there's some research that have a particular look at heavy metals, pesticides, mercury, manganese, etc. And they do show an increased risk for multiple system atrophy, and I think they play a role. And you could see that perhaps in somebody who has certain susceptibility for MSA, if you have additional environmental risk factors, that could really, trigger a disease process or exacerbate a disease process. But it's still something, we're learning about. And it's these environmental studies are often also quite tricky to do. They rely on recall memory of what have you been exposed to during your entire life. There's, it takes a lot of information. But certain things like pesticides do come up, and so they probably play a role. Same thing is true for well water drinking. Okay, and there's another question about gene engineering and gene therapy, which is a great question. I think a lot of the gene therapies are really, you need to know what the genetics is before you can develop gene therapies. So it's still very limited. So at this stage, the major genes that we have, we need to still understand how exactly they are related to the disease process before we can actually give it to patients. But a lot of the technologies exist now, and in related fields such as Parkinson's disease, there are gene therapies trials ongoing that, for example, try to lower the level of expression of synuclein, or that try to boost the protein handling machineries. And so I wouldn't be surprised if some of those trials are really helpful, that they may also be beneficial for potentially for the multiple system atrophy patient population. But I think we need to understand a lot of them a little bit more before we go right into the clinic. So this is where we're at the moment, we're still studying them in model systems. Okay. And so we are, are you recruiting for a clinical trial? So we are, so at NIH, we're just, one research center that does research in multiple system atrophy. There's a whole network of clinical centers, the research centers of excellence, that are recruiting patients on an ongoing basis across the country. So it's best to look for if you're interested in research, to look for a trial site that is close to you, that may be collaborating with us, because I don't think it's just one site that does the research. It really needs to be multiple sites working in parallel at various different approaches. And so at NIH, we mainly work on the basic science side. We try to understand what the molecular problems are, and colleagues at the universities are really more focused on bringing new therapeutic targets right to the patient. And so this is an ongoing process. So please check out the clinicaltrials.gov database for sites that are recruiting patients. Okay. So yeah, and there's some question, with, what is the actual timeline for when when we will actually have a drug that can slow the disease progression. It's very difficult to say, because while we are learning, it's very difficult to predict that in particular. But I also want to say there's a lot of hope. Our knowledge has really been accelerating. It's not just a linear growth, it's really been an exponential growth. We're also getting much better, not just on the genetic side, but also on the disease modeling side. And so there's reason to be hopeful that, perhaps we'll have really more targeted interventions within the next decade. And but this is research. We don't know, but we are hopeful, and we need patients to work with us and consider participating in clinical trials if you are affected. That may allow us to learn about this disease in the broader context. Okay. There's one question that is asking about specifically iron accumulation in autopsy brains of multiple system atrophy, which is actually a very interesting question. We do see iron building up, not just in multiple system atrophy, but also in a whole host of other neurodegenerative diseases. And it may just be, a factor that is related to a lot of the cellular stress that is going on, where there is really a propensity to develop protein deposits, but also iron deposits. So we don't really know what the exact mechanism is, whether it's an innocent bystander, but I think it's related to essentially cellular stress at this stage. Okay. And so let me see, let me just make sure I am looking at, so great talk, the CoQ2 modification, Asian ancestry. If European familial MSA genes were to exist, would you anticipate it is the same gene or a different gene? And so most likely, based on our current knowledge, is that it's actually probably something different. There have been really a number of studies now really looked at the CoQ2 gene itself. There's no real mutations. There's no enrichment of mutations or risk variants in the European ancestry population. There could be some changes that are affecting only perhaps some cells. So that's something that hasn't really been studied. So I want to, leave that open. But there could be other genes that perhaps regulate this CoQ2 on a cell level. So while CoQ2 may be fine in the Europeans, maybe some of the interactors could be affected. And there is some evidence that suggests that the levels of CoQ2 are a little bit lower in brain tissue of MSA patients. So even in European ancestry individuals, whether that is, a major problem in European ancestry cases, we don't know, but it's at least, it's at least something that, will have to be studied in more detail. And perhaps some of the follow-up clinical trials will actually shed some light into that. Okay. And then, so do you think that chemicals involved in dry cleaning could contribute to multiple system atrophy? Which is a great question. So it's certainly, there is a concern that especially in Parkinson's disease, it may play a role. There's really a growing body of literature. I'm not entirely sure if that's the problem per se in MSA for all of the neurological diseases, but a lot of these chemicals do have a mildly neurotoxic effect. So it's probably the doses that makes the poison. It's very hard to say to which extent that's a major contributor for the disease. Okay. All right, and then so there's a question, since a person with psoriatic arthritis has higher chances to develop Parkinsonism, would an IL-23 inhibitor have a better chance to prevent, postpone, or make MSA less progressive? So that's a great question. So all of those questions are, are legitimate. The honest answer is I don't know. We actually have seen some evidence that certain autoimmune diseases, which affect the bowel, seem to be correlated with multiple system atrophy. I didn't actually show it, but this is based on genetic evidence. And so perhaps, anti-inflammatory strategies may actually be really beneficial. And so, I expect that we'll see more clinical trials that really target the neuroinflammation that is accompanying multiple system atrophy and perhaps still have an impact. It's difficult to specifically say that one of the, IL-23 inhibitors will be the magic bullet, but I think there's reason to be hopeful. Okay. So is aerobic exercise important for enhancing dopamine signaling for multiple system atrophy patients? So aerobic exercise, I think in general is beneficial for all sorts of things. If it's, particularly because of the dopamine signal and for other effects, it's hard to tease apart. But in general, people who exercise a lot, they do better. They have more reserves. There's also decreased risk for cancers. There's, increased vascular health. So in general, if people can tolerate it, aerobic exercise is one of the best things you can do for brain health in general. And so I would strongly encourage it, but it's probably, it's multifaceted. It's not just affecting the dopamine signaling. It has multiple knock-on effects on the whole brain metabolism. All right, so I hope I got through all of the questions and I didn't miss anything. Maybe Cassie, if I missed something, please highlight it. But otherwise,
I think you got them all.
We are at the top of the hour. I hope this was helpful information. I'm also happy to answer questions that may come up later. Please feel free to email me. So as I mentioned, this is a discussion of the research. We don't really know where it's going to lead us, but we are hopeful, and I hope shedding some light into our various strategies makes you feel a little bit more confident that the work that we do is really impactful. And I'm happy to answer questions that may come up later. Thank you very much, everybody.